computer.
Okay.
Okay.
Okay. Hello, hello. Thank you everybody for being here. 9 a.m. Oh, we got more people coming in. 9 a.m. on a upsettingly snowy Idaho April morning for Brenlee's master's thesis. I think you guys are going to see some really fun and cool organismal science today that's really setting the stage for other research. So this is like the first step, I think, in a lot of very interesting work. So Brenlee got her BS degree in geology from George Mason University in Virginia in 2017. When she was there, she started a relationship with people at the Smithsonian that ultimately resulted in her publishing a brand new species of whale in 2019. That species of whale is kind of cool because it was from a deep sea fall and then was like, you guys ever seen a whale fall in the deep ocean that is then eaten by a lot of invertebrates? So that was part of that paper, was the taphonomy, the way that whale was consumed. And then that special piece of that project meant that PBS Eons did a whole episode about Brenlee's whale, and she didn't know it was coming. All of a sudden on YouTube, there was her paper, and there's like professional PBS talking about her science. After her undergraduate degree, she spent time in Montana at Makoshka State Park, where there's very important outcrop of the end Cretaceous and earliest parts of our era, the Cenozoic, so the dinosaur extinction, where she did a lot of outreach work. She then was a guest researcher in paleomammalogy at the Field Museum in Chicago. You'll hear a little bit, I think, about that maybe. And what's funny there is she did that with my postdoc advisor, Dr. Ken Angelzik, who's at the Field Museum. And so I ended my postdoc and Brenlee started her guest researching ships in the night. We didn't see each other in Chicago. But then a little ways in, applying for graduate school, Idaho State would be a great fit for Brenlee to continue this work that she started in Chicago. So she came here to do that master's. While Brenlee's been here, she has taken part in a lot of things on campus and in the department. And I think one of the ones you're going to be the most struck by is she took Ashley Rasmussen's biological illustration class. And you will see that this slides that Brenlee was able to artistically create pieces of work that help her science speak even more loudly. And so I think I'll stop for now, and I will let Brenlee tell you all about her research.
All right, excellent. I'm going to turn down the lights. Any objections? No, sounds good. All right. So thank you all for coming. I am Brenlee Shipps. I'm a candidate for the Master's in Biology here at Idaho State University and part of Dr. Brandon Peacock's Vertebrate Paleontology Lab. This is my thesis, The Topography of Diet, a comprehensive analysis of turtle triturating surface morphology using orientation patch count. Nearly all of my specimens were sourced from the Field Museum in Chicago and the Idaho Museum of Natural History here in Pocatello. Most of these specimens are from North America. Both of these institutions were founded on land cared for and occupied by indigenous peoples who also call these places home today. This work serves to honor the land by seeking to better understand the animals calling it home now and in the past. Turtles have and continue to be considered sacred symbols of the earth by many peoples around the world. I acknowledge the inherent dignity of these animals and am grateful to those who have worked to protect them and their homes. Turtles are among the most easily recognizable animals on the planet and play important roles in human culture. Depictions of turtles date back to at least pre-dynastic Egypt, and the idea that the world sits on a turtle's back can be found in Hindu, Chinese, and Native American creation stories. Yet despite their cultural ubiquity, a lot of what we know about turtles we've learned in the past 10 years. For example, just last year, we learned that turtles saw a threefold increase in speciation across the Eocene-Oligocene boundary 34 million years ago, when temperatures cooled and sea levels dropped worldwide. It seems odd that turtles, which are reptiles, would diversify when sea levels dropped and when temperatures dropped, but lower sea level meant more coastal habitat where turtles tend to thrive. Not until 2013 did we learn that turtle embryos construct their shell from their ribs as opposed to using part of the dermis. And though the origin of turtles was and still is a hotly debated subject among evolutionary biologists, molecular analyses completed over just the last decade show that they are most likely sister to crocodiles and birds. But despite how much we've learned about turtles, there is still a lot we don't know about the second most distinctive part of their anatomy, which is their beak. Turtles, like birds, have a keratin-based beak or ramphotheca. What we're seeing here are ventral or underside views of two skulls, a bald eagle and a red-eared slider. The highlighted skull bones are the premaxilla in green and the maxilla in blue. These are the keratinous beaks of both taxa. You'll notice that while the premaxilla and maxilla form the bony beak of these animals, the structure and organization of the bones and the shape of the keratin are different. Bird beaks are primarily an extension of the premaxilla, while turtle beaks are a sheath covering both the maxilla and premaxilla. The unique structure of bird beaks is highly derived compared with other beaked animals. So most examples of beaks in the fossil record more similar to the maxilla-dominated beaks of turtles than they are to the premaxilla-dominated beaks of birds. These include ceratopsian dinosaurs like Psittacosaurus, which is an early relative of Triceratops, Triassic non-dinosaur reptiles like the herbivorous Rhynchosaur Hyperodapedon, and the synapsid dicynodonts like Dicynodon, which are more closely related to mammals than anything else on this slide. Mom, you gotta mute yourself. Many of these fossil taxa have both a beak and teeth. Teeth are great for understanding one of the most fundamental aspects of an animal's biology, its diet. One of the most common outreach activities I did while working for Montana State Parks was show young visitors the difference between carnivorous and herbivorous teeth. Even little kids can see that cat teeth are sharp and cone-shaped, while cow teeth are flat and rectangular. But there's a lot more to the relationship between tooth shape and complexity and the diet of an animal. And with the increasing availability of high-quality 3D modeling software, we can get into much more detail about this relationship. Teeth can be analyzed using quantitative morphometrics. Quantitative morphometrics are methods that quantify the shape and form of a structure. And one of the most exciting examples of quantitative morphometrics is orientation patch count, or OPC. OPC is a measurement based on techniques for quantifying the complexity of a landscape. So a mountain range with a lot of peaks, like the Grand Tetons, would have a high OPC value, while a single peak, like Mount Rainier, would have a low OPC value. Multiple studies over the last decade have used OPC to measure the complexity of an animal's teeth. These two examples are from Maelstrom 2017, and the colorful shapes on the bottom are essentially topographic maps of the animal's teeth if you were looking directly down at them. Each color represents a different orientation or slope. Everything in pink is the same slope, everything in brown is the same slope, and so on. OPC measures complexity by counting how many faces or polygons exist on a model, then counting the number of groups that can be made of polygons that face the same direction. Each group of polygons with the same orientation or slope are called patches. This collared lizard's tooth is therefore cone-shaped and not terribly complex. The common chuckwallas tooth, though, shows something very different. While the collared lizard's tooth looks like one peak, the chuckwallas tooth looks more like a range of peaks. As a result, the collared lizard has a low OPC value, while the chuckwalla has a high OPC value. That's pretty cool on its own, but what's really useful about this technique is that it can be used to predict an animal's diet. A little kid can tell you that cone-shaped teeth probably belong to a carnivore, but OPC gives you the actual quantified complexity of a tooth. Previous studies have shown that more complex teeth indicate a more herbivorous diet, while less complex teeth indicate carnivory. The low OPC collared lizard is a carnivore, and the high OPC chuckwalla is an herbivore. Thus, we have a way of quantitatively measuring the topography of an animal's teeth in order to predict its diet. Once again, we are looking at the ventral or underside of several skulls. This time, we are only looking at turtles. During the late Triassic period, 220 million years ago, turtles had teeth. They had teeth on the maxilla, premaxilla, vomer, and pterygoid. Over the next 30 to 40 million years, turtles first lost the marginal teeth of their max and premax, then the palatal teeth of their vomer and pterygoid. Since the early Jurassic, 180 to 190 million years ago, turtles have been toothless. Instead of teeth, turtles have a hard keratinous beak. The part of their beak that interacts with food, the part where Odontocheles had its marginal teeth, is called the triturating surface. This surface is essentially a turtle's teeth, but it has never been quantitatively analyzed or comprehensively looked at. That means that we have no hard data on how this surface signals diet or phylogeny. Over the past 2.5 years, I have closely examined the beaks of 32 species of turtles to perform the first large-scale study of turtle triturating surfaces. I will present this to you in 4 parts. First, I will tell you about my specimens and how I determine their dietary categories. Second, I will answer whether the same pattern of increasing tooth complexity and herbivory found in teeth can also be found in turtle beaks. Third, I will compare the visible triturating surface of turtle beaks by family and diet. Finally, I will conclude by discussing future avenues for study and where this analysis should go next. First, let's talk about specimens and turtle diets. I mentioned that this is the first comprehensive analysis of turtle triturating surface, and that's true, but it doesn't mean no one has ever noticed or looked at this anatomy. 155 million years ago in the late Triassic, turtles split into the 2 suborders still around today. These are the Cryptodira, represented here by the red-eared slider on the right, Trichemi scripta, and the Pleurodira, represented by Irwin's Australian snapping turtle on the left, Alcea irwini. Cryptodira are the hidden neck turtles, meaning they pull their head directly backwards into their shell, as you can see when this red-eared slider tries to eat a strawberry. Pleurodira are the side neck turtles, which pull their head to the side under the edge of their shell rather than straight back. All of my specimens are cryptodirs because all of my specimens come from North American museums. Pleurodires are restricted to the southern continents, so they are less common in North American collections. What's kind of ironic, though, about this specimen diet bias is that the only other research on this surface was done on the aforementioned Pleurodiran Australian snapping turtles, genus Alcea. While researching on my own, I found no descriptions of what anything on this ventral surface is called. I asked Dr. Chuck Peterson here at ISU. I asked Dr. Jeanette Weinekin at Florida Atlantic University. I even asked the good people of the r/tortoise subreddit, and no one knew what parts of this anatomy are called. But most importantly, what I really wanted to know was the name for this ridge here. I saw this ridge on every turtle specimen I had and some of my pond turtles, but I didn't see it on any snapping turtles or box turtles. It is such a visible and distinct structure, but I didn't find anything. During my time here at ISU, I presented this research at a number of conferences, including Evolution 2021, Society for Vertebrate Paleontology, and SICB. It was while presenting at the Turtle Evolution Symposium that I finally got my answer. During one of the question and answer sessions at this conference, I asked the Zoom attendees if anyone knew what this structure was called. I got my answer from Scott Thompson, who is a researcher at the Museum of Zoology of the University of Sao Paulo and curator of the Chelonian Research Institute in Oviedo, Florida. Thompson sent me 3 of his papers on Australian snapping turtles, in which he used this surface to differentiate species within the genus. I finally learned that this structure is called the alveolar ridge. I will elaborate on the alveolar ridge more in part 3 of this presentation, when its presence or absence will be the major point of discussion. But for now, let's talk about the cryptodirs that I had. My specimens represent 7 families of cryptodiran turtles. I have 1 softshell turtle, 1 wood turtle, and 1 mud turtle. I have 4 sea turtles, 4 snapping turtles, and 11 tortoises. My data set is dominated by emetids, also known as pond turtles, because they are the most diverse clade of turtles in North America. Most of the iconic pet turtles that you see videos of online are emetids, including sliders and box turtles. I have 55 specimens total. 42 of these specimens have both skull and Rhamphotheca preserved, meaning that 13 specimens were only skull material without keratin. In order to determine if there is a signal between beak triturating surface complexity and diet, I needed to assign each species a dietary category by finding studies that reported what their wild diet consisted of. This most often meant studies that collected fecal samples from wild turtles, but some of them used stomach contents acquired from esophageal flushing or isotope analysis. Finding these studies was easy for some of the more charismatic taxa, like red-eared sliders and alligator snapping turtles. But some of these species, like the false map turtle and the pancake tortoise, had one or fewer studies examining their wild diet. In cases where data was scarce, I used Turtles of the World by Ernst and Barber, which was written in 1989. When I found more recent compilations of turtle species that reported diet, they always referred back to this book and the studies included in it. As far as I was able to find, the most recent book like this is in French and was written in 2006. There have been a lot of studies on turtle diet since this book was written, but I couldn't find any compilations of this newer data. I also reached out to zoos and reptile stores, but apparently captive turtles live off of pellets, mealworms, romaine lettuce, or in a few unfortunate cases, dog food. One of the outcomes of this thesis will likely be a publication of this diet review because it was a lot of work and no one should have to do this for their own project if I already have. So using what data I could find, I assigned each species 2 dietary categories. One based on what constitutes 90% of their diet and one based on what constitutes 60% of their diet. I created these symbols based on what represents most of the results that I found for each category. So for example, carnivorous turtles, it seemed like most often that meant they were eating a lot of fish. But omnivores here are represented by a container of omnivorous turtle pellets because I couldn't think of a more succinct icon for them. I've tried using a taco in the past, but that's like even less accurate. The 90% number comes from a standard set by Cooper and Vitt in 2002, and it determined that 90% gives an accurate report of the diet an animal is adapted for, while still accounting for variation in those diets. So these 4 categories on top, determined by what constitutes 90% or more of a turtle's diet, are what I'm going to call strict categories. However, I also wanted to know if omnivores that are more herbivorous had values similar to totally herbivorous taxa. And I also wanted to know that for carnivorous omnivores with carnivorous taxa. So out of curiosity and not following from any previous calculations, I also assigned each species a dietary category based on what constitutes 60% or more of their diet. By the strict definition of dietary categories, I had 32 omnivores, 7 carnivores, 13 herbivores, and 3 duraphages. Duraphagous here means that 90% or more of their wild diet is hard-shelled invertebrates, especially snails and crustaceans. I found all tortoises to be herbivorous, except for Chelonoidis carbonaria, which is the red-footed tortoise, and it is omnivorous. I also learned that a lot of tortoises consume feces as like a not insignificant part of their diet. And that's unfortunate from like a that's gross standpoint, but it is biologically interesting because it tends to be gravid females or females that are preparing to lay eggs that eat a lot of feces, implying that eating poop gives them the calcium they need to properly form eggs. Under the loose definition of dietary categories, 2 carnivores, both loggerhead sea turtles, were reassigned to mostly duraphagous. 6 omnivores, mostly Western box turtles, terrapin ornata, were reassigned to mostly carnivorous, and 7 omnivores were assigned to mostly herbivorous. This 60% or loose analysis therefore included 3 mostly durophagous specimens, 7 mostly herbivorous specimens, 6 mostly carnivorous specimens, 5 carnivorous, 13 herbivorous, and 18 omnivorous specimens. This adventure in turtle diet literature had a few surprising results. I already mentioned the omnivory of the red-footed tortoise, which I wasn't expecting because I'd assumed all tortoises were herbivorous. But even more surprising, at least to me, were the results that I found for the common snapping turtle. Snapping turtles are apex aquatic predators that have a reputation for biting off fingers. It stands to reason that sea serpentina are carnivorous, but their stomach contents always contain aquatic plants, and Ernst and Barber consider them omnivorous. Oresco and Gunsberger even found snapping turtles whose feces contained only aquatic plants. So even using the loose or 60% dietary categories, common snapping turtles are actually omnivorous. Once I determined the dietary categories, I needed to digitize my specimens so they could be analyzed by OPC. I created 3D models of each specimen's bone and keratin beak using photogrammetry. Photogrammetry involves taking a bunch of pictures of a subject at multiple angles, just like this animation here, which is made of an actual collection of photographs for this specimen, a red-eared slider. And these are all photographs that I took. Then I put all of these photos into Colmap, which compiles those images to create a 3D mesh of the specimen. The mesh that Colmap creates is not a watertight mesh, meaning that there are holes in it. Once I had this mesh called the fused mesh, I took it into a program called MeshLab to finish the model. This involved cutting the fused mesh to just the triturating surface, then creating a watertight Poisson mesh. Finally, I downsampled each mesh to 10,000 faces to account for differences in resolution between models and cut each mesh in half lengthwise to be more in keeping with OPC analyses of tooth rows. With every model complete, I could move on to the actual quantitative analysis of these structures, which brings us to part 2 and the first of my major questions. Does the same pattern of increasing complexity with increasing herbivory found in teeth apply to toothless beaks? This analysis uses the molar package for R created by Pampush et al. This process involved a lot of trial and error to generate OPC values that adequately captured the complexity of each surface without reading too much noise. What you're looking at are the topographic maps of several specimens. The images on the right do not correspond to the images on the left. This is a mix of specimens. Some use one side, some use the other. As I explained before, each color is a different slope or patch. These big black patches that you see are actually collections of smaller patches that were too small for R to include in the orientation patch count. Orientation patch count, the way that I used it, needed each patch to account for at least 1% of the surface of the mesh. So again, those black patches that you see are actually a lot of smaller patches. The actual value that I get from R is the orientation patch count rotated, or OPCR. Orientation patch count, or OPC, is the number of patches on a mesh where adjacent faces are oriented the same. Orientation patch count rotated, or OPCR, is a slight modification of this. OPCR calculates OPC values for a specimen 8 times, turning at 5.6 degrees for each calculation. The final OPCR value for a specimen is an average of all 8 rotations. This means that OPCR is better able to account for slight variations in the orientation of the mesh itself. I then compared bone and keratin OPCR values to see if there was any significant difference between the two. The reason I did this is because the actual triturating surface of a turtle's beak is the keratin, because that's the part that actually processes food. So if you think of your own head, it's not your skull bones that process your food, it's your teeth. Keratin doesn't preserve very well in the fossil record. And apparently a lot of collections destroy, discard, or do not separate the rhamphtheca from the beak when preparing turtle specimens. So the first step was to determine if the complexity of the bone of a turtle's beak is comparable enough in complexity to the keratin to use just the bone to predict diet. I did not find a significant difference between bone and keratin complexity, which is good because it means bone captures the actual complexity of the keratinous beak. That means that the keratin is essentially the same shape as the bone it sits on. But that being said, there are differences in how bone and keratin relate to each other by dietary category. This is a linear discriminant analysis showing the relationship of bone complexity to keratin complexity by diet category. Herbivores occupy a space in the middle of the graph, which means that they tend to have fairly average bone and keratin OPCR scores compared to duraphages and carnivores. Duraphages have high bone complexity, and carnivores tend to have low bone complexity. Omnivores occupy a big shape across the middle of the graph, but that's what you would expect to see because they're omnivores. It should overlap with the other categories. When we look at this data again using the loose or 60% dietary categories, we see a few interesting patterns. That big light green triangle is mostly herbivorous omnivores. It's a varied group that includes several ammidids as well as the red-footed tortoise. The mostly carnivorous omnivores, which are that pink polygon right up in the kind of top left, those plot in their own space entirely with high keratin complexity but lower bone complexity. This group is dominated by the Western box turtle, Terrapene ornata. We see this split between mostly herbivorous and mostly carnivorous omnivores when we look at just the bone too. In these jitter plots, each dot is one specimen, and the line in the middle of each rectangle is the mean for that category. In the strict dietary analysis that uses only 4 categories, omnivores sit pretty comfortably in the middle of everything else. But if you look closer, you'll see that the distribution of specimens within the omnivore category is split. There actually aren't any specimens close to the mean value for omnivores. And when we use the 60% categories in the top right, or your left, we see that the bimodal distribution of omnivores is driven by the difference between mostly herbivorous and mostly carnivorous omnivores. Mostly herbivorous omnivores are tied with duraphages for most complex bone. Looking at these results again in a histogram makes the overall conclusions clear. These categories are in order of increasing carnivory. Using the strict dietary categories, herbivores and omnivores are not significantly different from each other. But duraphages are more complex and carnivores are less complex than both herbivores and omnivores. The loose categories reflect this same pattern, but now we see that the high complexity of mostly herbivorous omnivores drives the omnivore values higher overall. What's really, really cool about these results is that they are very similar to results from previous studies that used orientation patch count to measure teeth. Carnivores generally have less complex food processing surfaces than any other group, whether you are looking at teeth or beaks. Santana et al.'s 2011 study of bat teeth found that frugivorous bats have much more complex teeth than omnivorous and insectivorous bats, while Mellstrom's 2017 study of extant reptiles showed that carnivores have much lower teeth complexity than herbivores, but that other categories exist somewhere in the middle. This study of turtles finds that carnivorous taxa have lower triturating surface complexity than any other dietary category. This study also finds that mostly herbivorous omnivores have significantly higher bone complexity than mostly carnivorous omnivores, and that duraphagous turtles have particularly high bone complexity. It appears that mostly duraphagous carnivores, so that's that light blue bar, that they have lower bone complexity, but there's only 3 of them in this data set and 2 of them are loggerheads. So it could just be that loggerhead sea turtles have particularly simple beaks that skew the rest of their category down. Finally, when we look at the same data organized by family, we see that tortoises have slightly more complex bone than ametids, but significantly more complex bone than sea turtles and snapping turtles. This makes sense given what we've already seen. Chelydrids or snapping turtles are either omnivores or carnivores, which likely makes their bone less complex. Cheloniids are skewed down by the loggerhead sea turtles, C. caretta, which are mostly duraphagous carnivores with particularly simple triturating surfaces. After mapping this data, I decided to throw an Ice Age snapping turtle into the mix to see where it would land. This specimen is Chelydra serpentina, which is the common snapping turtle of today. The white stars on this graph show my living chelydrids, and the black star is this fossil specimen. One of the extant chelydrids has particularly complex bone, but the fossil specimen fits right in with the other 2 in this data set. I would be very interested in seeing where other fossil turtles land, but this was the only one I happened to have made a photogrammetric mesh out of. With that, we head into part 3. What anatomical adaptations of triturating surface correlate with diet? This will be a more qualitative assessment of my specimens, with particular attention paid to the presence or absence of the alveolar ridge. When I started this project as a guest researcher at the Field Museum, I was particularly interested in specimens that had both keratin and bone. Out of the over 6,000 turtle specimens at the Field Museum, only 30 had both bone and keratin dry, intact, and easily separated. This slide shows you most of those specimens. It was immediately clear that this set of parameters was going to make finding specimens difficult. After finding as many as I could from museum collections, I decided to add 13 specimens that only had skull material, no keratin, because I already had access to them. This brought the total number of specimens to 55, but the number of specimens with both skull and ranthotheca to 42. Studying the alveolar ridge made me feel like a mad woman. I can't express how bizarre it is to see a structure on a bunch of these animals that seems to have a clear relationship to one of the most important aspects of their biology and yet find no information on it. But here it is, clear as day. Not only can you see this ridge in the gopher tortoise on the left or your right, but not in the snapping turtle. And you can also see the difference in keratin shape between these 2 species. The snapping turtle's keratin is rugose and it's heavily layered, but it's not organized. The keratin of the gopher tortoise is highly complex and highly specific. These ridges serve a purpose. Did you guys know that there is a genre of YouTube video that is just tortoise eating ASMR? I watched a lot of these videos. This is rhino. I am pretty sure rhino is a sulcata or African spurred tortoise, but the YouTube channel just says tortoise. While I unfortunately don't have a sulcata in my data set, it's unfortunate because they're actually my favorite tortoises, you can see that rhino clearly has that same ridge. If you watch closely, as he pulls back and as he opens his mouth, you see his bite marks leave behind the shape of that ridge. Every tortoise that I have looked at has this ridge on both their keratin and their bone. This is a selection of my tortoises with the alveolar ridge circled in each. It is definitely easier to see in some genera than others, but every one of them has it. This tracks with the overall high OPCR complexity values of tortoise beaks compared to those of other turtle families. There is a bit of an outlier. This is Malacochersis tornerii, the pancake tortoise. Pancake tortoises are small tortoises from sub-Saharan Africa. Adults are only about 7 inches long. They are elusive because that flat shell allows them to hide in thin crevices between rocks. They are also critically endangered and severely understudied. I did not find any dietary analyses of the pancake tortoise. Ernst and Barber considered them herbivorous, as most tortoises are, but they have the lowest beak complexity out of any tortoises in my data set, and their alveolar ridge is visibly reduced in every figure I saw of their skull, as well as in my specimen. Unfortunately, it's impossible to say what's going on with this animal until we learn more about it. While they're difficult to find in the wild, there are quite a few pancake tortoises in zoos. So a study of what foods and diets are most nutritious for them, even in captivity, would be an important step in understanding these little enigmas. But not only does every tortoise in my data set have an alveolar ridge, every herbivore in my data set has an alveolar ridge. It looks a little different depending on the taxa, but even green sea turtles, C. mitis here, have a distinctly raised portion on their maxilla. Only one obligate carnivore in my data set has this ridge, and none of the duraphages do. Mostly herbivorous omnivores have this ridge, but not mostly carnivorous omnivores. It is present in both cryptodires and pleurodires, because despite their name, the Australian snapping turtle that I mentioned earlier is actually an herbivore. At this stage, the alveolar ridge appears to be the result of independent convergent evolution for herbivory in disparate groups of turtles. This distribution raises the possibility of future studies on the evolution of feeding adaptations in beaked taxa. So if the alveolar ridge is a possible adaptation for herbivory, why does Eretmochelys imbricata have one? E. imbricata is the hawksbill sea turtle. It is fully carnivorous, but it is a weird carnivore. Hawksbill sea turtles are the only tetrapods, meaning they are the only non-fish vertebrates that are spongivorous. Their diet is at least 72 and up to 95% sponges. And sponges are mostly made up of silica. Silica is the primary ingredient in the production of glass. Plants also contain silica, and the grinding teeth of many modern mammals are an adaptation for dealing with this mineral. But even herbivores that eat high silica plants are totally outmatched by hawksbill turtles. While plants are as much as 20% silica by dry mass, sponges are up to 67% silica, meaning hawksbill sea turtles consume more than triple the amount of silica eaten by grazing mammals. This picture is dried intestinal contents from a hawksbill, and these little white sticks that you see are basically shards of glass. Given that we see this alveolar ridge in every herbivore, as well as this fungivore, it could be that the alveolar ridge is an adaptation for processing silica-rich food. But the alveolar ridge is also likely a signal for E. imbricata's ancestry. There are 6 extant chelonians: the green sea turtle, the loggerhead sea turtle, the hawksbill, the flatback, the olive ridley, and the Kemp's ridley. Both of the ridley sea turtles here are represented by the one Lepidochelys sketch. And if you were wondering where the leatherback sea turtle is, it's way over here in a different family. Of the chelonians, so everything connected by that light blue line, an alveolar ridge structure can be seen in the green hawksbill and flatback sea turtles, but not in the loggerhead or Ridley turtles. And in case you were curious, the leatherback doesn't have one either. Future research should address whether the alveolar ridge is present in the common ancestor of all chelonians. If it is, its presence in E. imbricata is likely advantageous to a silica-rich diet, not unlike the highly complex chewing teeth of grass-eating mammals. Though individual populations vary, Chelonia mitis, the green sea turtle, is generally herbivorous. And Natator depressus, the flatback sea turtle, is carnivorous with a preference for soft-bodied invertebrates. The only two chelonians that do not have an alveolar ridge are the Duraphagus loggerhead and Ridley. Again, future research on the distribution of this ridge in fossil turtles would be beneficial to better understanding the signal between ridge presence and diet. But I don't think it's terribly hard to imagine why a delicate ridge would be disadvantageous to a diet that is primarily made up of hard-shelled invertebrates. We see a similar pattern in Ametids. These are two subfamilies within the Ametidae, and both are represented in my data set. On this side, on this slide, terrapene represents one of these subfamilies, the Amedinae. None of the Amidines in my data set have an alveolar ridge. But within the other subfamily, Dairochelinae, Pseudemes, Chrysomes, and Trachomes do. The only two Dairochelinians without the alveolar ridge are the two Duraphagous taxa, Malaclemes and Graptomes. Which means that in both sea turtles and pond turtles, we see an alveolar ridge in all but Duraphagous taxa. At this stage, my data set suggests that the alveolar ridge may have evolved convergently in multiple groups of turtles, but that it was also lost convergently within these groups when taxa developed Duraphagous diets. This distribution raises the possibility of future studies on the evolution of feeding adaptations in beaked taxa. Okay, so now let's summarize what we've talked about so far. First, turtle diet has not been comprehensively collated in English since 1989. Next, we do not see a significant difference between keratin and bony beak topography overall, though the relationship between them varies with diet. We also saw that, just like in previous OPCR analyses of teeth, herbivorous turtles have more complex triturating surface topography than carnivorous turtles. Finally, we discussed the convergence of the alveolar ridge as an adaptation for herbivory in at least 3 families of turtles, and noted that the loss of this ridge may be a convergent adaptation for duraphagy. But I did not set out to study turtles. I love them, don't get me wrong, but the original motivation for this study was a very different group of animals. This is a dicynodont, specifically it is Diictodon, and unfortunately it is no longer with us. The last dicynodonts met their end in the late Triassic period about 201 million years ago, and none of their direct descendants roam the earth today. But when they were around, they spent most of their time being the dominant clade of terrestrial herbivores. Dicynodonts, especially later ones, had beaks instead of teeth, and likely had a keratinous ramphotheca covering their maxilla and premaxilla. This is a trace of a very bad picture of my boyfriend, William Primuth, who is about 6 feet tall, so he's going to be our scale here. And here's our friend Diictodon again, which is roughly the size of a dachshund. There were even smaller dicynodonts like Cysticephalus and Neacidon, who could probably fit in a purse, and much larger dicynodonts like Lysowitia and Endothiodon, who would need a big yard, lots of space to roam. Dicynodonts survived the worst mass extinction our planet has ever seen, the end Permian. And despite their bizarre appearance, they are the cousins of every mammal alive today. Most importantly, they are cute and I love them. However, no one loves them as much as Dr. Kenneth Angielsick, who was my supervisor when I worked as a guest researcher at the Field Museum in Chicago. Dr. Angelsic was the one who noticed that the enigmatic but evidently very functional beaks of Dicynodonts were strikingly similar to the beaks of turtles, despite the two groups not being particularly closely related. While at the Field Museum, we made meshes of a couple of Dicynodonts so we could test whether orientation patch count could predict diet from triturating surface complexity in them as well as in turtles. These are those meshes cut to just the toothless beak. I cut each one further so I could include it in a data set with my turtles, which means I cut each of these to be the same approximate shape as the turtle meshes. These overlays are ultimately what I cut each specimen down to. Again, I did this to try and get them as close to the same shape as the turtle meshes so I could compare them directly. These were the resulting OPCR values for each specimen, but let's put them into context. Dicynodonts don't really fit any particular category over the others, though the high complexity specimens are absolutely outside the range of carnivores. But these specimens span different size ranges and about 20 million years of time. So it's not exactly like fair to put them in an analysis with extant turtles. If you really wanted to understand how Dicynodonts plot, you need to compare a lot of them with each other. This quick analysis is essentially running on the presence of what if these were turtles? So to really look at this in Dicynodonts, you need to have a lot more Dicynodonts. But of course, Dicynodonts are not the only animals you could apply this technique to. Now that we know this method works on turtles, we can use it to analyze other beaked animals in the fossil record. This includes ceratopsian and hadrosaurid dinosaurs, brachiosaurs, pterosaurs, and maybe even fossil turtles. Of course, many of those animals have teeth in addition to beaks, but OPCR could absolutely be used on both the beak and the teeth to get more holistic diet information. There have been no other large-scale studies on the triturating surfaces of turtle beaks. This thesis serves as an introduction to the subject and a synthesis of what little data existed before. As this topic has had remarkably little focus even among turtle researchers, this study inspires more questions than it resolves, but that's not necessarily a bad thing. Turtles are fascinating animals, unlike anything else alive today. Despite their unassuming nature, their unique anatomy and evolutionary history make them a window into the past of life on Earth. By studying them, we improve our understanding of their diversity, inform decisions on conservation, and continue to build our knowledge of what came before us. This study involved a lot of collaboration for something done almost entirely during a pandemic. I'd like to thank the lovely folks at the Field Museum and the Idaho Museum of Natural History who helped me with specimens and software. My undergraduate intern, Farania Hung, Dr. Kristen Lane for letting Farania and I use her lab and being very patient with us, Scott Thompson, Dr. Sir Joshua Evans, and Dr. Jeanette Weineken for turtle-related advice. My committee members, Dr. Charles Peterson, Dr. Catherine Turner, and Dr. Leif Tapanilla for their advice, support, and in Chuck's case, camera. My lab mate Xavier and the rest of our cohort's very chaotic group chat, Dr. Kenneth Angelsic and Dr. Keegan Melstrom for their kindness, support, and knowledge, and my advisor, Dr. Brandon P. Cook, for everything. I'd also like to thank my family, friends, students, and coworkers, especially my kid sister Kaylee and my beloved partner Willie. But most importantly of all, I want to thank my bearded dragons, Loki and Bindi, whose consistent indifference to my every anxiety is both comforting and humbling, and whose company was much appreciated during quarantine. Thank you all for listening, and support your local turtles.
All right. Any questions for Brenlee? I can operate the chat and stuff.
Oh yeah.
Step out.
Yes.
Question.
Okay. Okay.
So given the low complexity solution, isn't that why the diet is more homogeneous?
Is more homogeneous.
Yeah, so like meat is just like
Sure.
That actually there's a greater diversity from the herbivores than the omnivores.
Yeah, so the question was, if carnivores tend to have relatively simple triturating surfaces, doesn't that imply that their diet is more homogeneous than the diets of omnivores and herbivores? And I would say that that's probably true. In terms of texture anyway, meat is meat. There are obviously slightly different textures, although I don't eat meat, so I wouldn't actually know, but there are different textures involved in eating meat, but not as different as there are if you are eating cactus, grass, lettuce, poop. There's a lot of different textures involved in an herbivorous diet and obviously in an omnivorous diet. So having a more complex beak topography may allow you to break down more diverse foods. Does that answer your question? Okay. Yes.
So beyond just beak structure and teeth structure, organisms, gut morphology process, that way is also important for determining what they can eat. So I know that's beyond the scope of your study, but are there any, are you aware of any research on extant species of turtles that correlates with their lymphatic intestine, or some characteristics like that that matches the structure?
So the question was, there is, I've talked about the differences in triturating surface morphology in diet, but what studies have been done on the gut morphology, right? And how the length of the gut or the microbiome of the gut can affect diet. And so there have been studies on that. I looked at some of them as I was doing the dietary categories. I remember being very surprised because a lot of animals that eat plants, particularly herbivores, do a lot of food processing, and birds that eat plants have a gizzard. So mammals chew, birds have a gizzard, turtles don't have either. So most of the work of breaking down food from a turtle comes from the fermentation in their gut. So that was kind of the extent of what I'd seen, but I know that there have been studies looking at the efficiency of breaking down plant material, depending on what type of setup you're using, whether you're using fermentation, teeth, or a gizzard. And I know that the teeth and gizzard are just as good as each other at breaking down plant material, or they're just as efficient. But I don't think that they'd actually looked at tortoises in the study that I read. So it's something that I'm very interested in, and it was definitely a question that came up as I was doing these dietary categories, but that's about all I got for it. Does that answer your question?
Yeah.
Okay.
I think you answered it well. It is important for organic understanding how their gut will,
Absolutely.
especially as you're trying to.
Yes, definitely.
We've got a Zoom question from Dr. Weineken. She asked, what populations of green turtles and hawksbills did you use?
Yes.
As their diets can differ substantially with population.
Okay, let's see if I can remember which ones they were. I know that I was able to get the specific population data for my green sea turtles and for the loggerheads. I don't think I had exact population data for the hawksbill, but both of the green sea turtles, I want to say, were from Baja, California. So I used studies specifically from that population and specifically looking at that group of turtles and that geographic location to assign the dietary category for my green sea turtle. And I think the loggerheads were both Caribbean. So I used, where was it from? It might've been from Trinidad, the study that I used to assign dietary categories for the loggerheads.
You're getting some nice compliments in the chat. All right,
Excellent.
another question from the chat from Ashley Ferguson. If
Hi,
the
Ashley.
herbivorous taxa have a ridge
Mhm.
and they consume silica-rich food, I'm
Mhm.
paraphrasing Ashley, why do you think the duraphagous animals do not have it, since some of the animals they're consuming have arachnidic shells or hard shells?
So One of the things that I didn't get to put in this presentation that I really wanted to was a video of a hawksbill sea turtle eating sponges. And I couldn't put it in because the frame rate was way too high and it just tanked the rest of the presentation in terms of like file size. So there are some really cool videos where you can see hawksbill turtles actually like kind of chewing for a turtle. Tortoises don't even really chew, but the hawksbill sea turtle really has to break down that material. It takes a couple bites, you can see it kind of munch. It actually spits the sponge back out, takes it back in, crunches on it some more. Sometimes it spits it out a third time. There is a lot of processing done to break down that silica. Now, when we had started this study looking at Durophagous taxa, we had expected their bone complexity to be really low because it doesn't seem like you'd need to have very high bone complexity if you're just crushing shells. But their bone is actually really rugose. So it doesn't have an alveolar ridge, but it is really well vascularized. So while Durophagous taxa don't have the alveolar ridge, they do have a lot of blood vessels underneath the keratin that probably allow them to feel nerve endings and blood vessels that probably allow them to get blood flow through that ridge better. But I don't think I actually answered the question. Why do you think they do not have it since they consume animals with aragonite? I think that it has more to do with the fact that if you are crushing something really hard, it's maybe less than ideal to have something relatively delicate on your triturating surface. It's easier to break it. And that's not great from like a living perspective. So I think that the Durophagous taxa lack this ridge because what they're doing is really crushing up and breaking shells, whereas what the spongivorous and the herbivorous taxa are doing is, it's not quite grinding, but the processing that happens is not as like, what's the right word? The impact of it isn't as high. Does that answer the question?
Ashley, are you satisfied?
Are you satisfied, Ashley?
She says yes.
Yeah.
Any other questions in house? We got the Zoom has got some. Kristen's got one.
Yes. So I know that the bite force for
Question was
turtles-
about bite force.
Oh, yeah, sorry. The question was about, have I found any structures that allow you to evaluate bite force? I have not. Other people have. The musculature of the turtle's jaw is really well studied because it is different in plurideers versus cryptideers. And so how that muscle connects has actually been looked at quite a bit. And the bite force of turtles has been looked at quite a bit. There's a lot of things about turtle feeding that have been really well analyzed, just not this, which is odd. So like one of the things that's also surprising about snapping turtles, they have a reputation for having a really strong bite, but it's actually not that strong compared with other things that have a high bite force. I don't remember what the value is, but it was lower than you expected it to be. Yes.
Josh has a question.
Thinking about those Durophagus a little bit.
Yeah.
Are there high complexity
Yeah.
thinking about other taxa, are there other taxa where that's been shown that there's really high complexity for crushing?
Yeah. So what's interesting is,
Question was about why
Sorry,
Durophagus have such high complexity.
yes. Yeah. So why did Durophagus have such high complexity? And are there other studies that show that? So I'm going to go back to, ooh,
Just click
I'm
on
just, can I close it?
Yeah. Arrows should work.
So I'm going to go back to, all the way back to the graph comparing study and other studies. Okay, so you see from here that Keegan's 2017 study did not necessarily find that Durophagus have particularly high complexity in teeth. I don't know how much I'm allowed to say, but I know Keegan said that there is some preliminary data from another study he's doing that may be showing this. So it is an ongoing area of research. Most of the work using OPCR is from the last decade. So I think that it is in progress.
If I can add one
Yeah,
thing,
absolutely.
one thing that's really interesting is teeth and a keratinous beak are very different in terms of like what the tissue is. And so a lot of gyrophagous tetrapods, their teeth are like little spheres.
Yes, yes.
So very simple. Whereas the bone of a keratinous beak, the bone supporting a keratinous beak that's crushing is extremely complex as a surface because of all of the like attachment structure for that beak.
Yes.
So there's that kind of a thing to think of too.
But then the overlying keratin is not necessarily very complex.
All right, one last question to be sensitive of time. Turtles famously tend to live a long time.
Yes.
Do you know of any taxa that undergo shifts in diet through ontogeny? What might we expect to see or not see in
Yep.
feeding related morphology as a turtle grows up?
So this was a major, okay, so you all heard it because you just read it.
Yeah.
So this was a major part of looking at turtle diet determination, because there are a lot of turtles that change their diet through ontogeny. For example, green sea turtles are always thought to be herbivorous, but young green sea turtles are mostly carnivorous. And I think that's true of sliders as well. So there's a lot of turtles where they have more flexible diets when they're young, and then that diet becomes less flexible as they get older. What I tried to do as I was looking through the diet categories was use data that was mostly on adult turtles, but if I did have a particularly small skull, then I kind of considered what the juvenile data was in addition to that. So that's why some of these categories, rather than being like one of the strict herbivore or carnivore or dirophage, they ended up in an omnivore category because I just didn't know how old some of them were. But I'm not totally sure, but it is a really interesting subject of how does that triturating surface actually change as the animal ages? Does it change a lot? Do you see, like, do young green sea turtles not really have those little bumps on the beak, but older ones do? It would be really cool to look at, though.
All right, let's give Brenlee another hand.
Way to go, Brenlee.
Guys, Zoom, feel free to unmute and yell whatever. you want.
Great job, Brenlee. Love the artwork.
Thanks, Mom and Dad.
There we go, get the grid going.
Yeah, Brenlee.
Very cool, thanks everybody.
How many people ended up being there?
I don't know. We'll be delicate about all this process.
21 or 22?
What?
21 or 22? You had quite a crowd.
The Zoom, yeah.
Oh, thank you.
Sorry about the volume again, Brenlee.
That's okay. It was really funny.
I think I did that last time too.
I think you did, but that's okay.
Stop recording.